Okay, in this next section, I'm going to be talking about histones and looking at histone modifications and how they affect gene expression via compacting chromatin. So firstly, this is a structure of how DNA wraps around histone proteins. So DNA is there shown in yellow, and histones are proteins. There's 8 of them. Histone 2A, 2B, 3 and 4 that form a histone optimer. And as the histones sit there with DNA wrapped around them, there are N-terminal histone tails that protrude outwards. And these are shown here. Now these tails can be acetylated or methylated at very specific amino acid residues. And these are what we call histone acetylation and histone methylation. And to cut a long story very short, acetyl groups generally make the DNA wrap around the histones much more loosely. The methyl groups, most of them, cause DNA to wrap around the histones very tightly, and tight wrapping of DNA around histones prevents transcription factors from binding. So this is just a little bit more detail on histones and themselves. So this is the form that chromatin takes. You've got DNA wrapped around nucleosomal cores. Each nucleosome is made up of 2 each of histone 2A, 2B, 3 and 4. So that's a histone optimer. There's about 147 base pairs of DNA between each histone core. And this is just taken out of a textbook showing how you can separate out the DNA from the histones. But basically, if you were to degrade this linker DNA, you would find 147 base pairs of DNA wrapped around histones and about 50 base pairs in this linker region. And this is consistent across all different forms of chromatin, whether it's euchromatin or heterochromatin. And then if you dissociate the octameric cores, that's where you get your 8 different proteins out of there. So although this structure is consistent throughout chromatin, it's how tightly packed these nucleosomes are. And this slide shows the effect of histone modifications on how transcription factors can bind to the DNA. So here's DNA wrapped around the histone cores, and what happens is the DNA temporarily can unloop and then rebind. And this happens, in a fraction of a second. It can unloop and it can rebind. Whilst it's unlooped, a DNA binding protein such as transcription factor can nip in and bind to its crucial region. So this could be estrogen receptor, this could be the estrogen response element, and whilst it's exposed, the transcription factor can gain access to this DNA and then bind in with the DNA. And this would be an example of a transcription factor binding to a specific DNA sequence. So what you can see is if the DNA remains tightly bound, the transcription factors can't get in. Equally, if the histones are compacted together very tightly, then transcription factors cannot get in. And histone acetylation promotes this loose chromatin. Most methylations promote the DNA being tightly packed around the histones. So this is controlled by a series of enzymes. There's the histone acetyl transferase, which adds acetyl groups, whereas histone deacetylases can remove them. Now this can be although these are heritable upon cell division, they also can change as the cell differentiates. Now most methylated histones are associated with repression of gene expression. A minority of these are associated with gene activation. So the general way of remembering this is that DNA methylation is gene silencing. Most methylations of histones are gene silencing. Acetylations are activating. You don't get acetylation of DNA, only get acetylation of histones. Now these histone methylations and acetylations are heritable upon cell division, much like in the way that DNA is. DNA, you've got 2 strands that separate. Well, with your histone cores, the nucleosome cores, you've got 2 histone 2Bs, 2 histone 3s and so on. So when DNA replicates, these can split down the middle, and if you've got a methylated histone there, you'll have a methylated histone on the other histone H3, and the two can separate, and that allows the two separate strands to maintain the histone methylation pattern during DNA replication. So this slide shows what the chromatin is like under the different situations. So up at the top we have got acetylated histones, particularly histone H3, which we'll talk about fairly shortly. That's associated with active transcription. So histone acetyltransferases will promote that, whereas histone deacetylases, HDACs, will remove acetyl groups. So you go from this configuration to the unmodified configuration, which could be active, it could be non-active. But the important thing is, if you want to go from being the acetylated to the methylated, you have to remove the acetyl groups, and that's what HDACs do, histone deacetylase enzymes. Once the histones are unacetylated, they can be methylated. So this is where your histone methyltransferases will tag these N-terminal histone tails with methyl groups, and then they will recruit in a group of general heterochromatin proteins, which will cause the histones to become tightly packed together, and you'll see there's no opportunity for transcription factors to gain access to the DNA in this formation. This is transcriptionally silent chromatin. Much like you see at the telomeres and in those heterochromatin regions, there is no chance of transcription factors gaining access to this DNA. Whereas it might be able to gain access here, it will be nice and loose here. So the next thing to look at is, what are the actual sites of histone modifications? Now, the ones that really regulate gene expression and heterochromatin are on histone H3. And histone H3, the N-terminal tails that stick out is this region here, and this illustrates all the different places where these histones can be modified in some way. Now, the most important ones are histone H3K9, so that's lysine 9, which can be methylated, and that will cause heterochromatin formation and gene silencing. And the way we would write this is H3K9ME. So histone H3K9, which is lysine 9, and then ME for methylation. And that can be ME3, as shown down here, so which is where 3 methyl groups are on this free NH3 group. So this is lysine, and there's an NH3 group there, and each of those hydrogens can be replaced with CH3 group, and that will give you your lysine K9Me3. And it will look like this, and that will recruit in those heterochromatin proteins. Alternatively, it can be acetylated. So this is where the acetyl group is tagged on there, and that is associated with gene expression. So histone H3K9AC is the code that we would write for the active gene expression configuration. Now, confusingly, histone H3 lysine 4 methylation is also associated with gene expression. That's the only methylation you'll ever come across that's associated with active gene expression. Ignore the phosphates. Here we've got lysine, histone H3, lysine 14, acetyl acetylation, that's active gene expression. And then the other major one, which is histone H3, lysine 27, ME3, is associated with X chromosome inactivation and silencing of Hox genes, which we're going to come on to later. These are Hox genes are developmental genes, very, very important in tissue patterning. So this slide shows the importance of histone H3 lysine 27 trimethylation. Now, this particularly regulates Hox genes or homeobox genes, which are involved in embryonic patterning early on in development. And these genes are incredibly well conserved in evolution, and the same gene that controls the ends of the anterior and the posterior of the fruit fly will control the same bits of the same respective bits of a human, as close as you can possibly get. And this is because organisms are designed to develop embryonically, starting at the head and ending at the posterior. So what happens in both humans, fruit flies, and all living things that have got a linear body pattern, so that's basically all animals apart from jellyfish and associated things, will start off in fruit fly, the lab gene will be switched on, and then the proboscis, and then various abdominal, thorax and abdominal genes will be switched on in sequence. So this gets switched on first, which gives rise to the head and the mouth parts, and then these blue ones will be switched on, which will give these parts, and so on. So this gets switched on first, which helps to switch on this gene, which helps to switch on this gene, which helps to switch on this gene. So this helps to pattern the organism, but this switching on and switching off is all controlled by histone H3 lysine 27 trimethylation. Now, a technical term for this is spatial and temporal collinearity, which is just a clever way of saying that this gene cluster, and these are in a cluster, this gene sits next to this, which sits next to this. This gene physically gets switched on, and then this gets switched on, and then this gets switched on, but then by the time this one is on, this one here has been switched off. So basically there is a run of transcriptional activity down the chromosome, starting here, working its way down to here, and by the time we're down here, the lab and the proboscis genes are being thoroughly silenced by histone H3 lysine trimethylation. Now, exactly the same process is going on in humans, the difference being that we have got multiple clusters of Hox genes all doing the same thing but controlling different aspects of human development. So I'm now going to go through some other examples of epigenetic changes that are occurring in cells and tissues that can affect cellular behavior that is down to an epigenetic phenomenon. And the first one of these is neutrophil extracellular traps. Now, neutrophils are white blood cells, and they have this clever ability of when they sense lipopolysaccharide from bacteria, they can very quickly loosen their chromatin and then eject it from the cell, and the DNA is effectively spewed out of the cell, and that DNA is very sticky to bacteria. So where you've got a site of infection, you've got a lot of bacteria there, neutrophils will home into that area, they will then change the chromatin structure, and they will effectively explode, spewing out their DNA in something called a neutrophil extracellular trap. That whole process requires the remodeling of the chromatin, and that chromatin remodeling involves histone H3 and as having a specific arginine residue. Arginine, like lysine, has got an NH3 group available to be modified, and that becomes citrulinated, which is a post-translational modification, by this enzyme peptidyl arginine deaminase 4, or PAD4. So that citrulinates histone H3 and allows this DNA to be kicked out of the cell. Whereas if you have cells which are PAD4 mutants, that are PAD4 deficient, they cannot do this. And if you create a mouse model lacking in PAD4, then it cannot form these nets, so the cells can't undergo NETosis, and then they have an impaired immune system. Now, this same enzyme, PAD4, also has an important role in citrulinating other histones as well, in this case histone 1. Now, histone 1 almost acts as a sticking plaster to hold down the DNA when it's wrapped around the histones. So you can see here, these are the histones, tightly packed histones, and there's histone H1 there. Now, when this is citrulinated, and this is not in neutrophils, this is in stem cells, when histone H1 is citrulinated by PAD4, again, this decondenses the chromatin, allowing transcription factors to gain access to genes that they otherwise wouldn't be able to gain access to. So here we've got PAD4 citrulinating histone H1, helping histone H1 to become removed. That allows RNA polymerase to bind to some of the master regulator genes which control stem cell behavior. One of the key ones is NANOG, another one is KLF2, and these genes are master regulators which control stem cell behavior. So again, it's modifying the chromatin so it is super loose. These genes would normally be silenced in all differentiated cells, whereas if citrullination of histone H1 occurs, transcription factors can gain access to these genes which are normally silenced. Okay, so the next phenomenon where epigenetics is absolutely crucial is in X chromosome inactivation. So hopefully that you will all know that in females who have 2 X chromosomes, one of those X chromosomes is randomly and permanently silenced. And this results in the formation of what we call a bar body. And that is a silenced X chromosome there right on the edge of the nucleus. And this X chromosome silencing is down to histone methylation again. It's controlled by a gene called the X-ist gene. And the X-ist gene is expressed, very tightly expressed. In males, it's not expressed. In females, which are XX, it's expressed to a moderate amount, and that allows normal silencing of X chromosomes. If you have females with additional X chromosomes or males with additional X chromosomes, you get more X-ist RNA produced, and that will silence all but one of those X chromosomes. So the best example of X chromosome inactivation is the tortoiseshell cat. It's a nice animal model of the process. This link here is a really good link to Citable, which is a Nature publishing site which explains the whole process of X inactivation in mammals. So take a look at that site. But basically, what we've got here is a female cat which has inherited the black fur gene on one X chromosome and the orange fur gene on the other X chromosome. Now, because those are 2 different hair colors on the X chromosome, one of those X chromosomes is going to be silenced in every cell, but that is going to be silenced in a random process as the animal develops. So you end up with small patches of cells that express the black fur, the gene for black fur, and other patches that express the gene for orange fur. So you end up with small patches of skin expressing the gene for orange fur next to small patches of cells expressing the gene for black fur. So have a look through this for a fuller explanation, but this is happening in females, you just can't see it. If you take a patch of skin cells from a female, they will maybe silence the maternal X chromosome, and then a few cells along from that you'll get a patch of cells which are paternal X chromosome, which is active. Now this process occurs by the expression of the X-ist RNA, which binds at the X inactivation center. Now, exactly how this only silences one chromosome is very complicated and not fully understood. But what we find is we get the coating of the entire chromosome. So initially you get binding with the X-ist RNA to the X inactivation center, and then the RNA spreads down the chromosome till the entire chromosome is covered or coated with this X-ist RNA, and that silences one of the X chromosomes. So this is just shown in a little bit more detail here. You've got the X-ist RNA, which effectively covers the whole of the chromosome, initially starts off there and then migrates up the chromosomes. The end result of this is histone H3 hypoacetylation and then histone H3 lysine 27 trimethylation, and that results in the compaction of the X chromosome and silencing of all genes on that silenced X chromosome. And this is a process that occurs randomly early on in embryogenesis. And this is illustrated here. This is a slide using a fluorescent probe to detect the X-is transcript in an undifferentiated embryonic stem cell. So remember, embryonic stem cells are cells that can differentiate into any cell type. And here we have got two fluorescent regions which represent regions where the X-ist RNA is binding. And so this represents the two X chromosomes in this undifferentiated cell. Now both of these X chromosomes will be active at this point. Upon differentiation, one of those X chromosomes will accumulate X-ist RNA all the way down it. The other one has still got the X-ist center visible there. But then when you've got complete differentiation of that cell, the X-ist RNA is coating one of the X chromosomes, the other one is not visible. That X chromosome that's not visible will be active. So this is the silent one because it's covered in the X-ist RNA. The other one is the active X chromosome. Now this is the final part of the lecture, and this is looking at situations where epigenetics goes wrong and results in a disease situation. So here we're looking at a group of diseases called imprinting diseases, and the diseases that we're looking at are Prader-Willi and Angelman syndrome, and these are both caused by an imprinting defect on chromosome 15. Now if you remember right at the beginning, I told you that sperm DNA is actively demethylated and egg DNA is passively demethylated. And in those first few days after fertilization, almost all the methylations are stripped off. There's two regions, well, a small number of regions where that doesn't happen, and those are at the imprinting regions, where the maternal and paternal methylation patterns are maintained through that early process of stripping off the methylations. Now that's really important because in embryogenesis, some genes are only expressed from maternal derived DNA and some are expressed from paternal derived DNA. Now in this imprinting center on chromosome 15, there are a group of genes, these 6 are the main ones, and these 4 here are expressed from paternal DNA, i.e. the sperm DNA, and therefore these 2 genes here are expressed from the maternal derived DNA. So what should happen is that on one chromosome 15, these genes are expressed from the father's DNA and these are silent, and on the other chromosome 15, these 2 genes are expressed, but these 4 are silent. Now that normally works perfectly well. The problem comes is if there is a deletion of this region on either the maternal derived chromosome or the paternal derived chromosome. Now for most situations, if you lose a small region of DNA, it doesn't matter because you've got 2 copies of that DNA and the second copy will in many cases make up for the absence of DNA. So if you get a micro deletion and lose a few genes, often there is no phenotype to that. However, here there is a very severe phenotype because if let's say a sperm cell has got a deletion of chromosome 15q 11 to 13 and this whole region is deleted, yes, all of these genes exist in the egg DNA. However, these 4 genes here will not be expressed from the egg DNA, and therefore, as the fetus develops, these 4 genes will be absent, and that will result in the disease called Prader-Willi syndrome, and it's caused by the SNRPN gene here, which is the crucial gene that causes the disease. If this gene is absent from the paternal DNA, Prader-Willi syndrome will occur. If the egg DNA has a microdeletion across this region, then these two genes here will not be expressed from the paternal chromosome 15, and it's the UBE3A gene that's absolutely crucial. In the absence of the UBE3A gene, the fetus will develop Angelman syndrome. Now, this disease can come from these diseases can come from many different sort of genetic causes. The microdeletions are the main cause of them. So just a small bit of chromosome 15 is missing, and that causes most Prader-Willi and Angelman syndrome. However, you can also get a disease called the disease caused by uniparental disomy, which basically means that you have got a fertilized egg which has lost the paternal chromosome 15, but has compensated by duplicating the maternal chromosome 15. So you've still got two copies of chromosome 15, it's just that the two that you've got only express these two genes and not these four genes. So uniparental disomy can cause Prader-Willi syndrome, less of an issue, less common for Angelman syndrome. And then, because this gene is absolutely so crucial, if a point mutation occurs which is non-epigenetic, a point mutation in the UBE3A gene that inactivates the UBE3A gene will also cause Angelman syndrome. So these diseases have a central underlying epigenetic cause because of the imprinting. Now, the imprinting that occurs across this region, this histone H4 acetylation and histone H3 lysine 4 methylation. So in summary, epigenetics is not what we call the blueprint for a human. The blueprint is a set of instructions that allows the complete formation of a human, of something. That's what a definition of a blueprint is. The genetic sequence, all the A's, G's, C's and T's is not enough. You need the epigenetic information as well to allow, it's not just, what I'm trying to say is it's not just the DNA sequence, it's also whether those regions of DNA are actively, epigenetically active or inactive. Cellular differentiation is controlled by this process. So as we've seen early on in embryogenesis, you get active demethylation of sperm DNA, you get passive demethylation, you get methylation of CpG islands that affect gene expression. So initially you get demethylation and then remethylation of specific CpG islands, which would then allow cells to differentiate down different lineages. We also, so that's at the DNA level. At the histone level, we get methylation and acetylation. Acetylation is always associated with active expression. Methylation is not always, but most methylations are inactivating, a few are activating. And then we've covered various pathological and developmental processes where epigenetics is crucial.